Chapter 2. The Double Helix. This is Life. I got hooked on the gene during my third year at the University of Chicago. Until then, I had planned to be a naturalist and looked forward to a career far removed from the urban bustle of Chicago's South Side, where I grew up. My change of heart was inspired not by an unforgettable teacher, but a little book that appeared in 1944, What is Life? By the Austrian-born father of wave mechanics, Erwin Schrödinger. It grew out of several lectures he had given the year before at the Institute for Advanced Study in Dublin. That a great physicist had taken the time to write about biology caught my fancy. In those days, like most people, I considered chemistry and physics to be the real sciences, and theoretical physicists were science's top dogs. Schrödinger argued that life could be thought of in terms of storing and passing on biological information. Chromosomes were thus simply information bearers. Because so much information had to be packed into every cell, it must be compressed into what Schrödinger called a hereditary code script, embedded in the molecular fabric of chromosomes. To understand life, then, we would have to identify these molecules and crack their code. He even speculated that understanding life, which would involve finding the gene, might take us beyond the laws of physics as we then understood them. Schrödinger's book was tremendously influential. Many of those who would become major players in Act I of molecular biology's great drama, including Francis Crick, a former physicist himself, had, like me, read What is Life and been impressed. In my own case, Schrödinger struck a chord because I, too, was intrigued by the essence of life. A small minority of scientists still thought life depended upon a vital force emanating from an all-powerful God. But like most of my teachers, I disdained the very idea of vitalism. If such a vital force were calling the shots in nature's game, there was little hope life would ever be understood through the methods of science. On the other hand, the notion that life might be perpetuated by means of an instruction book inscribed in a secret code appealed to me. What sort of molecular code could be so elaborate as to convey all the multitudinous wonder of the living world? And what sort of molecular trick could ensure that the code is exactly copied every time a chromosome duplicates? At the time of Schrödinger's Dublin lectures, most biologists supposed that proteins would eventually be identified as the primary bearers of genetic instruction. Proteins are molecular chains built up from 20 different building blocks, the amino acids. Because permutations in the order of amino acids along the chain are virtually infinite, proteins could, in principle, readily encode the information underpinning life's extraordinary diversity. DNA, then, was not considered a serious candidate for the bearer of code scripts, even though it was exclusively located on chromosomes and had been known about for some 75 years. In 1869, Friedrich Miescher, a Swiss biochemist working in Germany, had isolated from pus-soaked bandages supplied by a local hospital a substance he called nuclein. Because pus consists largely of white blood cells, which, unlike red blood cells, have nuclei, and therefore DNA-containing chromosomes, Miescher had stumbled on a good source of DNA. When he later discovered that nuclein was to be found in chromosomes alone, Miescher understood that his discovery was indeed a big one. In 1893 he wrote, "Inheritance ensures a continuity in form from generation to generation that lies even deeper than the chemical molecule. It lies in the structuring atomic groups. In this sense, I am a supporter of the chemical heredity theory." Nevertheless, for decades afterward, chemistry would remain unequal to the task of analyzing the immense size and complexity of the DNA molecule. Only in the 1930s was DNA shown to be a long molecule containing 4 different chemical bases: adenine, A, guanine, G, thymine, T, and cytosine, C. But at the time of Schrödinger's lectures, it was still unclear just how the subunits called deoxynucleotides of the molecule were chemically linked. Nor was it known whether DNA molecules might vary in their sequences of the 4 different bases. If DNA were indeed Schrödinger's code script, then the molecule would have to be capable of existing in an immense number of different forms. But back then, it was still considered a possibility that one simple sequence like AGTC might be repeated over and over along the entire length of DNA chains. DNA did not move into the genetic limelight until 1944, when Oswald Avery's lab at the Rockefeller Institute in New York City reported that the composition of the surface coats of pneumonia bacteria could be changed. This was not the result he and his junior colleagues, Colin MacLeod and Macklin McCarty, expected. For more than a decade, Avery's group had been following up on another most unexpected observation made in 1928 by Fred Griffith, a scientist in the British Ministry of Health. Griffith was interested in pneumonia and studied its bacterial agent, pneumococcus. It was known that there were 2 strains designated smooth S and rough R, according to their appearance under the microscope. These strains differed not only visually, but also in their virulence. Inject S bacteria into a mouse, and within a few days the mouse dies. Inject R bacteria, and the mouse remains